you. So last up, we're going to hear from Denis Torre from the links, BD2K Links Data Coordination Center. This is a connection with the DataMed BioCaddy effort that was identified fairly early on and extends things into the area beyond the indexing and shows how indexing can lead on to other things.
Thank you, Ian. So, hello everyone, my name is Denis Torre. I'm here representing the Links DCIC, and today I'm going to introduce a pilot project we've been working on known as Datasets2Tools. So the overarching goal of a project is to facilitate the connection between biomedical datasets and the computational tools which can operate on them by introducing a new type of digital object known as the canned analysis. To promote the findability of this object, we aim to enrich DataMed and other biomedical repositories with interfaces which will allow users to access the canned analyses directly from within the dataset search pages and dataset landing pages of these websites. So I'll start off by giving a brief overview of the background behind the project, follow up by explaining in detail what a canned analysis is, and wrap up the presentation by giving a general overview of the interfaces we aim to enrich DataMed and other biomedical repositories with. So as we all know, there's a large wealth of biological information stored across multiple biomedical repositories online, and the BioCaddy DataMed indexes many of these, such as the Gene Expression Omnibus, the Nuclear Receptor Signaling Atlas, Links, PDB, and ENCODE. On the other hand, there is a wide variety of computational tools which can operate on these processed datasets to extract valuable biological knowledge. Many of these are indexed by tool repositories, such as Aztec Bio, which is developed by the BD2K CCC, or OMIC tools. So the use of computational tools on the datasets stored within the repositories is a key step in extracting valuable biological knowledge, which can increase our understanding of biological systems and ultimately drive biomedical research. However, none of the aforementioned repositories currently provide a method to systematically associate the datasets stored within them to the wide variety of computational tools which can operate on them to extract knowledge. So the key aim of our project is to develop a platform which will allow users to create, store, browse, and find associations between datasets and the computational tools which can operate on them. So the key question we ask ourselves when defining this project is how to define a simple but effective solution to linking datasets to the computational tools which can operate on them. So the solution we propose to address this issue is a new type of digital object known as the canned analysis. So in our model, the canned analysis is defined by three key elements. The first element is a link to a dataset which is stored on a biomedical repository online and is available to users of the scientific community. The second element is a link to a computational tool which can operate on that dataset to extract valuable biological knowledge. And the third element, an element we named canned analysis URL, is a hard-coded link to an external web page which contains the results of the analysis run by the tool on the dataset. Ideally, this will be provided by the computational tool itself. The object can be further enriched by adding a description of the analysis, which will allow users to better understand the aims, and any number of customizable metadata tags. So we believe this object has a series of properties which will make it a useful solution in addressing the problem of connecting datasets and tools. So first of all, the structure of its metadata is independent of both dataset type and computational tool type. This allows, on one hand, for accurate annotation of every single digital object, and on the other hand, also allows interoperability between datasets and computational tools coming from non-cooperating resources and platforms. Second, the canned analysis is indexed in a relatively simple relational database and can be therefore found and retrieved by performing queries on the metadata tags associated to it. The canned analysis is also easily shareable by providing the canned analysis URL, which can also be embedded as an icon within websites. So information about tools, datasets which can be analyzed by them, and the canned analyses will be stored in a database, which we aim to make accessible to users across the scientific community through a Google Chrome browser extension, a website, and an API. The Chrome browser extension will enrich the dataset landing pages and dataset search pages of DataMed and other repositories by allowing users to easily access canned analyses associated to each dataset. And I will give a thorough example of it later on. Whereas the websites and the APIs will allow users to find, create, browse, and share these analyses either through a user interface, in the case of a website, or programmatically, in the case of the API. So now I'll give a brief overview of the functionalities we aim to add to DataMed with the Google Chrome browser extension. So in this case, we can see a screenshot of a results page of a dataset search on the DataMed index. The functionality added by the Chrome extension is represented by the toolbars which are displayed below each search result. So in this case, upon loading one of these search results page, the Chrome extension scrapes the HTML content of a web page in order to extract the accession IDs of every search result, and then communicates with the Datasets2Tools database in order to extract all the canned analyses which are associated to a specific dataset, and displays them below each search result in the form of an interactive toolbar. So every icon here represents a different computational tool which has been used to analyze that dataset. Upon clicking on one of these icons, the user will trigger a drop-down menu which contains the tool's name, a table containing a list of all the canned analyses which are associated to the tool and dataset pair, and a search bar which allows users to filter the results. The first column, labeled link column, contains icons which link to the canned analyses URLs. So by clicking on one of these icons, the users are directed to an external web page by means of a hard-coded link which contains the results of the analysis run by the tool on the dataset. In this specific case, the link links to an external web page stored on the Enricher website developed by the Maya laboratory at Mount Sinai, which contains the enrichment results of the top 100 most overexpressed genes in a PGFR knockout versus control experiment of the dataset. The second column is quite self-explanatory, contains a brief user-inputted description of the analysis. The third column allows users to browse the metadata associated to the canned analysis object interactively by mousing over the small information icon on the left. And it can also allow them to download the metadata by clicking on the icon on the right. And we aim to add this functionality in a variety of formats, such as TXT, JSON, or Excel tables. The final column will allow users to share the canned analysis by providing a button which allows to copy the link to the URL to the clipboard, or by providing a brief snippet of HTML code, which will allow users to embed the link as an icon within websites. Finally, if a user wants to have more information regarding the computational tool which has been used to run the analyses, he will be able to mouse over a small information icon at the top. By clicking on it, he will trigger another menu which contains a brief tool description, information about the tool developer, links to the tool homepage, and if available, references and links to video tutorials. So this wraps it up for the overview of the functionalities we aim to add to the Google Chrome extension in terms of canned analysis findability. And of course, this model could be extended to other repositories other than DataMed itself. So in the last couple of slides, I will show a few models and examples of how this protocol could be extended to datasets, to repositories rather, other than DataMed. So this screenshot shows the results page of a dataset search performed on the Gene Expression Omnibus dataset page. And in this case, the functionality added is the very same I just showed on DataMed, and is represented by the toolbars displayed below each search result. And again, these display interactive links, icons, which can link to the drop-down menus and therefore link to the canned analysis results. This other screenshot displays transcriptomics dataset landing page on the links data portal. So this is still a model. This is, of course, being implemented and still open to discussion. In this case, the functionality added is displayed by the table at the bottom. In this case, every row is represented by a different tool. So we have information about the tool's name, a brief description, and on the right-hand side, the number of canned analyses which are associated to the tool and dataset pair. The table can also be filtered using the search bar above. So upon clicking on one of these plus icons on the right-hand side of the table, the user will be redirected to a second page which contains information about the tool and an interface with information about associated canned analyses, which is very similar to the one displayed in the Google Chrome extension. It will have links, descriptions, access to metadata, and sharing functionalities. In this case, we also display a model for a more refined search interface, which takes advantage of the metadata tags associated to each canned analysis. To wrap up, the final example, this is another model which could potentially show a way this platform could be implemented within the NURSA landing pages. This is a dataset landing page on the NURSA website. It's the dataset title. We have information about experiments, an overview, and in this case, the functionality is added in the canned analysis tab below. Again, we have a table containing information about tool names, with descriptions, and canned analyses. By clicking on the plus icon on the right-hand side, the users are directed to a second page contains information about tools and all the information regarding associated canned analyses. So overall, the aims of the Datasets2Tools project is to allow users to find canned analyses directly from within the dataset landing pages of DataMed and potentially extending this to other repositories. It will allow users to access associated metadata, allow users to share these canned analyses across the scientific community. And we ultimately hope that this model could become native to the DataMed website in such a way that it will allow users to access these functionalities without necessarily having to download the Google Chrome extension. So that wraps it up, so thank you for your attention.
Thank you, Denis. So one quick question, I mean, just to clarify, when you're talking about a canned analysis, is it an analysis that somebody has already performed on the data?
Yes, yes.
Okay.
So we aim to, so this was a brief overview of how this project would relate to the findability of these objects. So on the other hand, we also aim to provide forms and APIs which will allow developers of computational tools to register analyses on our database.
So if someone performs an analysis,
Yeah.
they can register it, but you're also discovering analyses that have already been performed?
Mm-hmm. So for example, a developer of a computational tool might automatically run analyses on a certain number of data sets within a repository, and we will provide the functionality to upload these links to our database so that these analyses could be discovered by users who also not necessarily have computational expertise.
Okay. All right. Thank you. Any other questions for Denis?
This is very interesting, and I wonder how actually flexible it is to be, for example, converted into a Dataset2Standards tool. So because you are searching the tool and they're indexed, and in BioSharing we index the standards, and also we would like to associate the standards to the relevant data set where the annotation is done. How flexible is your code? Is open and something we could collaborate and some other objects that were meant, object other than tools?
No, it's still very open to, we're still defining it, it's still in the works.
Excellent.
So I'm very happy to talk about how to extend it also
Excellent.
to standards as well as tools.
Cool, thank you.
You described the association between a specific data set and a tool. There is a similarity to workflow systems where it's not data sets, but it's basically an interface or standard that links then to Susanna's question. So on the data side, there might be a standard format which is implemented, in which the data is available, and on the tool side, an interface which knows how to deal with this data. This is how you can pipe things through standard workflow systems. Is this something you are, and then of course the next question is, in principle, then the relationship could be only between the interface and the tool, and what you now have in a static way could be dynamic and with a kind of caching system. So somebody decides, run this tool on this data set, and then you just cache the results. So is this something you are considering, or is this would this be too much development effort?
So the project is still in the works, so of course we're open to discussion about this. Our aim was now just to remain on the datasets and to initially enrich DataMed and hopefully see if this works. And if we find an agreement, we could then move on to expanding this to a more extended view.
Yep, one more.
Yeah, I guess the CAN analysis is a great idea. My question regarding to the tools and data sets, I think there's a, the gap and between tools and data sets regarding, at least you need to define a standard of the interface from both the tool side and data side. My question for you is there, are you planning to, propose this kind of a, idea to the public, or there's any difficult for you to incorporate any other types of tools into this framework.
So the idea behind the canned analysis is that it's a very simple object. We don't want to enforce any necessary structures. So until now, we're still defining it, of course, but it's designed to support any form of tool and data set. So we aim to make this as flexible as possible, as lightweight as possible, and we aim to make this a way to allow for interoperability between datasets and tools which come from non-cooperative platforms. So yeah, we aim to make this as extensible as possible in terms of resources and developers.